So welcome everybody to Phenotype February. We are now February 8th, which means we've got 7 conversations started and 21 more to go. For those of you who have not yet gotten engaged, or for those of you who have privately told me that you're very intimidated by what we're trying to accomplish, I'm going to try to provide an easy way for all of you to ease into participating in this community conversations that we're trying to start up. And hopefully raise awareness for those of you who haven't been paying attention to what we've been doing for this first week, and really actively inviting you all to join in this this community activity that we're trying to foster for the next month. So on our last community call, we kicked off Phenotype February, and on our forums, I also posted a description of what I discussed on our last week's call. But briefly, the idea of Phenotype February is that we recognize that phenotype development evaluation is a core fundamental activity that we all need in order to generate high quality, reliable evidence. And yet the science of phenotyping is something that we need to figure out how to be working on together. And rather than thinking about it as just another thing you do as part of a study, I really wanted us to try to focus some coordinated effort towards thinking about the problem of phenotyping and the issues that arise as a concerted effort. So we're using the 28 days of February to focus on methodologic issues about phenotype development evaluation. And we're doing that by showcasing 28 different phenotype targets, one a day, to hopefully have a community conversation about the methodologic issues and clinical issues about how to create reliable phenotypes in our community. On our call last week, we did a live survey to ask all of you what phenotypes would be of interest to you, and you all voted and picked some at the top of the list. And in the forum post, I just pasted the rank ordered list of the top vote getters, that all of the different phenotype targets that we actually could go after this month. As a community, our goal is just try to make progress on as many of these topics as we can. And so I didn't actually plan to do it in the way that it's played out, but based on the votes that you all had, I've been picking phenotypes this last week that are come from your votes, and have also invited others in the community to raise their hand to volunteer to lead a conversation about other phenotypes. And so we're going to just, as a community, try to make progress on this collection of phenotypes that you all felt were important areas for us to focus on. So as Craig mentioned, he's created a useful resource that if you haven't gone out or if you weren't aware of it, it could be just a helpful way to orient yourself to those who are trying to figure out where to join in the conversation. Out at odyssey.org/Phenotype February, you're going to see this page. And on this page, there basically is just our calendar of 28 days, 28 phenotypes, and where the chats are, where the conversations have started. I'm motivated last week by this idea that no matter what you are trying to do in the Odyssey community, even if you're really on the data side and just thinking about standardizing your data, or you're thinking about trying to generate evidence and thinking about some sort of characterization, estimation, or prediction design, no matter where you are in this conversation, if you aren't doing phenotyping, you're probably doing something wrong, because the journey to get to evidence always runs through phenotyping. And with appropriate pun intended, one of our key Achilles heels in our Odyssey community is the fact that if we can't create high quality phenotypes, we can't generate high quality evidence. And so while we've done amazing work with developing standards for converting the data to the OMOP Common Data Model, our vocabulary team's done amazing jobs creating appropriate mappings of source codes to standard concepts. We've got really groundbreaking statistical technology and machinery to enable large-scale analytics. But ultimately, our statistical processes for characterization, estimation, and prediction, they take as input phenotypes. And so if we pass in garbage phenotypes, we are going to get garbage evidence out, even if we're running the world's best statistics. Conversely, if we can actually come up as a community with developing high-quality phenotypes, it actually can be the building block that actually enables us to generate a wide array of different high quality evidence And it's not the kind of thing where everybody has to be an expert in phenotyping, but as a community, we really need to, my assertion is that we need to continue to focus on improving the science of phenotype development evaluation, as well as put those best practices into actual applied clinical practice by building phenotypes that we can actually use in our research. I talked about last week also that ultimately I would really like for us as a community to rally around the idea that we can build systems for generating reliable evidence. I'm really excited by Adam Black's leadership and Paul Naji's leadership with our open source community work group, because we need developers to think about how we can actually together build systems that generate evidence to get out of the current bespoke one-off custom approach that we go about generating evidence one drip at a time, and start to think about actually how we can actually scale the evidence generation process by making high quality steps everywhere along, from data quality evaluation, phenotype development evaluation, and analysis reliability evaluation. And it's this critical juncture of phenotype development evaluation where I think we are still maturing as a broader research community about how to do this. We know that lots of research done right now is using phenotypes that have substantial measurement error. We don't yet actually incorporate measurement error appropriately into our analysis. So there's a lot of work to be done. But the only way that we're going to really be able to realize this vision of building systems to generate reliable evidence is if we just tackle the pieces brick by brick to build that appropriate foundation. A phenotype process could look something like this. I highlighted last week that we can talk about the process of creating concept set expressions, so identifying the codes that represent the events that you want to look for. We can define cohort definition logic as it relates to initial events, inclusion criteria, and exit criteria. And we can use a host of different tools to help us evaluate our cohorts. And what I've been specifically trying to highlight over this last week of Phenotype February is how there's a lot to be learned at each and every one of the steps of this process. There are also a lot of different tools that our Odyssey community has built to support the phenotype development and evaluation process. And very purposely, one of the things I've tried to do over this last week has been to highlight and enumerate opportunities for different Odyssey tools to make a material impact in contribution to the phenotype development process. And so for those you haven't seen, and I'll go through examples here in just a bit, but we can make heavy use of Atlas as a tool to design cohorts. We do have a programming language in CAP-R that allows us to programmatically create phenotypes, and we'd like to showcase that. We have Phenotype as a recommender system for concept sets. We have Aphrodite as a tool to build probabilistic phenotyping. We have Cohort Diagnostics as a tool for evaluating phenotypes using population level characterization. Then we have PhEvaluator as a tool to actually do phenotype assessment and estimate measurement error based on a probabilistic model. And these are just examples of the types of tools that are in our community that touch on the problem of phenotype development and evaluation. But my assertion, or I think what I've observed across our community is not everybody has the same level of familiarity with all these tools. And so I think there's an opportunity for us to learn about these tools and think about how they could be applied to the particular problems we might be interested in. So I'm going to say this now, and I'm going to state it at the end too. Phenotype February is supposed to be a community activity where you all engage. How do you engage? You join a conversation. I've been trying very hard to start conversations on the Odyssey forums by posting my own random musings about different clinical ideas and different methodologic aspects about this. And so I've been posting new threads each and every day, starting Phenotype February. Craig created that very nice resource. If you're like getting confused about how to look through all the forum posts, there's one place at odyssey.org/Phenotype February where you can see where the starting point of each of the conversations is. But I'm not posting the answers to these problems. I'm starting a conversation that I want you all to engage in. I'm providing a motivation for what is a clinical idea we might want to phenotype, and then providing perspectives of some angle that I've just arbitrarily chosen to try to show some reflections of something interesting that I find interesting about that phenotype as it relates to applying our Odyssey tools. But I'm also making all of those tools publicly available. So when I create a phenotype definition, I'm posting them publicly so you can go explore them yourselves. When we've been using our tools and generating results, we're sharing the results so you can go review the results yourself. And what I really encourage all of you to do is read and reflect on the conversation and then join the conversation. So reply to the threads, post your own thoughts and reflections. If you see something interesting or want to challenge the stupid things that I've said, feel free to post and become part of this conversation as we learn from each other about how we develop phenotypes and also hopefully evolve towards reaching some sort of level of consensus that we've got a phenotype that seems like it might be reasonable for use in specific contexts. Another opportunity for you all to engage, for those of you who have data, we are creating phenotype definitions, which means you can run them against your data set. And that can be as simple as just executing the cohort against your data and seeing how many patients you actually find. And that can go as even further to say, why don't you run cohort diagnostics as a standardized tool to allow you to evaluate the phenotype locally within your own environment. And if the mood strikes you, you can actually share the results back with us, and we'll be able to actually post them out on data.odyssey.org. Gautam has been doing a great job of, as I've been creating these phenotypes, he has actually been executing them against data we have access to within Janssen and posting the results on data.odyssey.org. So each of the 7 days we've got, or at least 6 of them are already out there, you can actually see how different databases perform against all those phenotypes that we have been playing with. But to encourage others to join, both because you'd have the opportunity to learn about how to actually run a cohort definition in your environment, whether you're using Atlas or otherwise, also to learn how to run the cohort diagnostics package. There's a nice vignette that kind of describes the step-by-step instructions, but if you haven't actually done it, this can serve as a useful forcing function to get you going on there. And whether you just choose to do that locally and just, share your insights of saying this is what I've seen, or whether you want to share your results, this is an opportunity for you to build that phenotyping muscle and develop this competency for yourself. And then I also want to encourage, for those of you who get somewhat motivated by this or would like to join in, you have an opportunity to lead a discussion. So I told you last week that I was going to own the responsibility of starting 7 conversations. I have now done that. And that means that there's an opportunity now that anybody who would like to lead a different conversation on a different phenotype, you are welcome and encouraged. I'm excited that we've gotten a bunch of volunteers who have picked their topic and their date. And I'll show you what days are available. But for those that are interested, there still are plenty of open days available in Phenotype February if anybody is interested in learning how to in leading a conversation and sharing what you've learned, and hopefully getting the opportunity to learn from others about a phenotype target that is of interest to you. So anybody who would like to lead a conversation, feel free to just reach out to me at ryan.odyssey.org or on the Odyssey MS Teams environment and just say, hey, I'd like to lead something, and then I would love to collaborate with you to lead that conversation. All right, so with that all said, one more thing. We've also created a whole bunch of Phenotype February resources that are going to be useful. There is an Atlas instance, atlas-phenotype.odyssey.org. This is a Atlas instance we are using as part of our phenotype development and evaluation work group, but this is the place that I'm posting all of the phenotype or cohort definitions that we are creating thus far. And so if anybody's interested in actually going seeing those in practice in Atlas, you actually can go see all of them right now. And I'll show that to you in just a second. If you haven't yet applied for or just filled out this Google form to get access, it's just a security enabled thing. So you just need your email so that we can give you an account so that you can go log in there. And on the Odyssey forums, there's the link to this link that I'm also sharing on the screen here of how you can just request access. Another resource that we've created is an instance of cohort diagnostics. It's out at data.odyssey.org/phenotype February. And this is where we are posting all of the cohort diagnostics results for all of the phenotypes we've been created throughout the days, and we'll continue to do that. And I think that that's a great place if you're just interested in like learning more, just being able to explore the results that are out there and see what you learn and talk about what you reflect on what you've learned by looking through the data. I think that can be a great way that you can participate and contribute. There is a Odyssey GitHub repository for Phenotype February. You can actually just download the Git repo, and that will basically allow you to run cohort diagnostics in your own local environment against the Phenotype February definitions. And so if you're interested in running that just for your own experience, then that's a great thing to do. And if you want to share the results from there, those results could just basically be directly shared, and then they would be posted out on data.odyssey.org. Again, it's only containing aggregate summary statistics, so there's no issues about data sharing that's going on here. And last but probably most important is our Phenotype Development Evaluation Work Group is a great resource for all of you. Gautam and Aza led a very productive office hours last week. We had several folks come and ask questions both on the technical side of, like, I saw John just posted, like, how does one run cohort diagnostics? So someone came to the work group office hours and asked that specific question, and Gautam helped folks debug their local environment to make sure they could run cohort diagnostics. And someone also came with a clinical question and said, well, how do we actually phenotype delirium? And Aza walked through the process of actually developing and evaluating a phenotype for delirium, and that'll be showcased actually later this week for the work that happened literally just last week together at office hours. And so I encourage you all to use our community as the primary resource to talk to each other, learn from each other, share your experiences, ask the questions that you don't know answers to, and also respond to those questions that you see coming from other members of the community. Together, we can make a lot of progress if we collaborate in this space, but it actually requires you all to roll up your sleeves and actually contribute, not just be passive lurkers watching as I scream from the rooftops to myself. All right. With that said, here's our calendar of Phenotype February. I committed that I was going to do 7 days to get us started, and indeed you will now see conversations started on type 2 diabetes, type 1 diabetes, atrial fibrillation, multiple myeloma, Alzheimer's disease, hemorrhagic events, and neutropenia. I'll talk a little bit about my shared reflections as I've started those conversations in a little bit. What you're going to see is we got a lot of exciting phenotypes coming down the pipe. So I want to thank Aza, Joel, Gautam, Asya, Rupa, Tiffany, Juan, Anna, and Alan, who have already raised their hand and volunteered to take a day to cover another phenotype. You'll see there are still some blank days here. If anybody else is interested in leading a conversation about a phenotype, just reach out to me. Otherwise, you'll be subjected to me posting my own rambling thoughts, and I've heard from multiple members of my Janssen team that they really hate when they see one of my very long posts because they feel some obligation that they might have to read it. So save my team some heartache and actually contribute by leading a phenotype yourself. But I'm excited to see all of the different, you can see across this range of things, we're seeing phenotypes that are being developed across pretty much all of the possible disease areas and system organ classes or therapeutic areas that we might be interested in. We're also seeing a range of different topics that are coming from the vantage point of methodologic aspects of what it actually takes to develop phenotypes. And so I'm really excited to see as we dive deep into these clinical areas and try to make substantive progress towards the phenotypes for these clinical areas, but also that we're using this as a forcing function to try to evolve our own understanding of the phenotype development and evaluation process, and hopefully learn from each other about tools and tips and practices that maybe can evolve into some sort of consensus best practices that we can all learn from and follow. All right, so with that, I'm going to give you a 10-minute whirlwind session through 7 phenotype conversations that I've tried to initiate. This is only to entice you to hopefully go actually read the full conversations that have been going on. But for those of you who haven't yet read them and are just interested in like what is going on, I'll give you the quick run through. On day 1, we talked about type 2 diabetes. Here, the clinical idea is very clear. There's a chronic disease of type 2 diabetes that we've oftentimes studied for many, many different reasons, whether that be an indication for a wide array of treatments or also as an outcome that we want to study. And from a methodologic perspective, what I tried to showcase here is how we can actually go through the end-to-end process of developing a phenotype using Atlas, with using Atlas as a tool to develop the cohort definition, Phoebe as a tool to recommend concept sets, and then we used cohort diagnostics as a tool to evaluate the definitions that we created. In the case of type 2 diabetes, some of the interesting insights here is to think about diabetes as the broad classification, which includes different subtypes of diabetes, such as type 1 diabetes, type 2 diabetes, and secondary diabetes, and what that can actually mean as it relates to us trying to reach greater specificity and making sure that we really are finding our type 2 patients, not just anybody with broad scale diabetes. The other thing we highlighted here is the notion of index date misspecification. One of the things I highlighted as we talked about this is that if you're trying to find newly diagnosed patients, when is the index date of someone being newly recognized as diabetes? And what we saw is that the first time someone had a diagnosis, actually prior to that diagnosis, they might have already been treated with a type 2 diabetes drug, or they already had an HbA1c measurement value greater than 6.5. So they already had diabetes prior to the moment that they were first diagnosed. And so thinking about the methodologic issues about how do we create cohort definitions that actually have good index dates is an area that, if you're interested in that conversation, type 2 diabetes could be a good conversation for you to read about and join in the conversation. Day 2, I talked about type 1 diabetes. I was surprised that was one of the highly voted items for the group, but I decided to tackle that given that we had just done type 2 diabetes. And here, again, using cohort diagnostics, we had a very contrasting story. Whereas the type 2 diabetes definition looked generally pretty good and reasonable and matched our clinical intuition, the type 1 diabetes definition that I posited as a starting point actually looked like a horrible definition, because we would expect that most type 1 diabetics would be newly diagnosed maybe in their early teens or early 20s. We would expect that most type 1 diabetics would actually be treated with insulin. And yet what we actually saw in certain databases was a pattern that made it look like we actually had misclassification of type 1 diabetics who were being pulled into our definition even though they probably were type 2 diabetics. And so the issue of misclassification and what we might actually do about it became part of this conversation. Joel Swerdell added a bunch of really nice commentary on the end of this, using Phevaluator as an approach for us to quantify the measurement error. And it really highlighted opportunities for us to improve the phenotype development, the phenotype algorithm itself. And so if you're interested in those types of topics, I'd encourage you to take a look at day 2. On day 3, I talked about atrial fibrillation, highlighted that there were several papers that have been published very recently trying to create phenotypes on atrial fibrillation, and notably pointed out that some of them actually had different codes to define the same clinical idea. Here I wanted to showcase specifically the process of how you could take a published paper with its own set of local source codes. Oftentimes in the US, those would be papers published with an ICD code list, an ICD-9 code list, or an ICD-10 code list. And I walked through the process of how you could actually replicate an existing definition. So without regard to whether it's good or bad, just the idea that you want to produce what was done in a paper. Sometimes people misinterpret the OMOP Common Data Model and Odyssey vocabularies as something that basically says because you start with ICD codes, you can't use ICD codes as part of the phenotype development process. And nothing could be further from the truth. If you want to start with an ICD list, you absolutely can, and it can be a useful starting point to build a phenotype. And so in this conversation, I just wanted to start talking about how does one actually go about learning from an existing published definition, and then how can we actually implement that in our own environment. And so for those of you interested in atrial fibrillation or that process to discovery, feel free to check that out on day 3. On day 4, we dove into oncology and specifically looked at multiple myeloma as a disease target. This conversation was really to highlight that there are specific, in the oncology space, we often talk about that you really need a lot of specialty data to be able to reliably identify specific cancers, and that can be true for specific subtypes, but there's still an awful lot we can learn from general population data sets. And so in this particular case, I showcase how we can actually implement alternative definitions for multiple myeloma, again, looking at literature-based definitions that actually use procedures and treatments as part of the regimen, and we can actually see the impact of those choices. So check that out. On day 5, I highlighted Alzheimer's disease and specifically highlighted that there have been a range of different papers, and interestingly highlighting a paper that was led by Dave Kern and Soledad Cepeda in our community, which specifically thought of the problem of if you're targeting Alzheimer's disease, and Alzheimer's disease specifically is a phenotype that might have low sensitivity, meaning that you're missing some of the people that actually have Alzheimer's disease, maybe a strategy you could take is to actually create a phenotype for dementia as the broader idea and use that as a more sensitive variant of Alzheimer's disease. And I highlighted how you could actually create some of those implementations and think about the differences of there. So if you're interested in Alzheimer's disease or how to use multiple alternative definitions, this is a good chat to get a part of. Day 6, hemorrhagic events. Lots of people are interested in bleeds for lots of reasons. And yet it actually turns out that there's lots of very thorny algorithms out there for how do you kind of figure out whether or not a bleeding event is actually taking place. And so we examine a recent paper by Wayne Ray and colleagues that provides a pretty elaborate definition for how to create bleeding-related hospitalizations. We implement it in the Odyssey tools, and then we also evaluate whether or not we could create a more generalizable approach to that phenotype that would work in some data sets that are not just US administrative claims data. And so if you're interested in that topic, I'd encourage you to take a look at day 6. And day 7, yesterday, I posted about neutropenia. I saw Anna Scharfman was on the call for it. I know that she and Wojciech and others hate the fact that measurement data are noisy. And this is, of course, a problem that affects many of us who have data, particularly in electronic health record data, where even if we standardize to the OMOP common data model, that does not mean that we have one measurement concept for one clinical idea. It doesn't mean that we've normalized to one unit. And it doesn't even mean that we even know what the actual plausible range of values could be for that measurement. And yet the reality of it is data sets may have outpatient labs, outpatient diagnosis, inpatient diagnosis, and you might want to create a definition that tries to make use of any and all of that information to find a clinical idea. And so in this discussion, we talk about tools such as ARIES as an approach to use your Achilles results as a way to find what measurement unit pairs actually exist in your data, and to figure out what plausible ranges actually are occurring so that you can create a definition that handles the fact that measurement data are noisy. So that is 7 conversations that I've started. I would encourage all of you to join all of them, or at least take a look and participate in whatever strikes your fancy. We got 3 more weeks of Phenotype February to go. Those conversations are started. They are not ended. But we hope that you will all engage and participate in this dialogue, review the results, and share your own insights. Ultimately, at the end of this, if we can reach the point where we have a shared level of understanding and a shared level of confidence in phenotypes, we might be able to use these resources that we're creating as part of our process to generate evidence across all of our studies. But at a minimum, just getting shared awareness and hopefully increased confidence in the phenotype development evaluation process and the best practices that we're trying to evolve in the community. That if that's what we accomplished this month, then I would consider it to be a tremendous success. So I encourage all of you to join and engage in this conversation, and I encourage you to look forward to the rest of Phenotype February and all of the contributions that our community is going to make. And if anybody would like to lead any other parts of this conversation, then please reach out to me. I would love to have your support. I know we've got a few minutes left, so I'll shut up, ask if there's any questions, or otherwise let you go back to enjoying the rest of your Phenotype February.
Patrick, it's Christian. 2 questions, even there's no time. When you're saying we talked, where does the talking happen, or is it just in the forum?
It's all on the forums.
All on the forums. Okay. And question, so there's no actually audio, video talking.
So these weekly calls is where we're trying to highlight the points,
Okay.
as well as on the phenotype work group, the conversations can take place. But really, we're trying to make sure that we are supporting this on the asynchronous chat on the forums.
Cool. Second question, the results, right? So the results look like persons with atrial fibrillation per Wharton et al. 2021. Is there a, and then of course all the, the details, definitions and JSONs and so on. Is there a something of a clinical description or of a rationale and who Wharton is and so on and so on?
Yeah, so every one of those forum posts, if you read them, you'll
Okay.
see that I've authored a clinical description of the target.
Got
and
it.
then I've also tried to provide a context for the phenotype. So in this case, let's talk about the paper and that kind of thing.
Got it. Got it. That's the answer. That's the answer.
John, you can go ahead.
Yeah, Patrick, you mentioned office hours. I'm assuming that there's a link to that and a time for that in your slide deck, and that slide deck will be put up on the Odyssey site.
Certainly my slides will be shared. Actually, Azza, I see you. Do you have a specific time for the phenotype work group office hours this next week?
So next week is actually our regular meeting for the working group. So we are holding a regular meeting. We'll also answer questions. So it's a combination, the phenotype development and evaluation work group meeting and office hours. So I will post the actual invite so you can log in, and it will be on Teams. But currently we are holding every Friday 10 to 12 office hours for the group. So you can just join the phenotype development and evaluation anytime, Friday 10 to 12 through the rest of the month, and we'll answer
Friday 10 to 11 is the 1 hour I cannot miss this week, but I'll be there late.
11
I'll be there an hour
12.
late.
Okay.
Yeah.
We'll be
Just
there.
to clarify what Azza said, we alternate between office hours and the work group. So every other week is either the work every week is either the work group or the office hours, and it's always Friday 10 to 12.
We posted that form as well in the chat to get access to Atlas. I have seen a couple of people say they filled it out but didn't get access. Gautam, should they reach out to you if they have any troubles with that?
So there is a thread on the forum for requests. Lee Evans, our administrator, is monitoring it, and if there is any requests for about technical issues, he's been promptly responding on that thread.
Great.